oral histology has always been an intriguing aspect of dentistry. One of the most non-understood concepts in it is the process of odontogenesis or tooth formation. Today, we are going to look into the finer aspects of amelogenesis, also commonly known as enamel formation. The term basically encompasses the events taking place during enamel formation. We already know that enamel, by virtue of its high content of mineral salts and their crystalline arrangement, is the hardest calcified tissue in the human body. The function of the enamel is to form a resilient covering to the teeth to enable mastication. Dating back to the development of a tooth, we recall that the cells of the inner enamel epithelium are derived from the basal cell layer of the oral epithelium. Before enamel formation begins, these cells assume a more columnar form and differentiate into ameloblasts that produce the enamel matrix. The changes in the shape and structure of cells of the inner enamel epithelium are collectively categorized under the life cycle of ameloblasts. According to their function and morphology, the lifespan of cells of the inner enamel epithelium can be divided into 6 stages: morphogenic, organizing, formative, maturative, protective, and desmolytic. A very interesting way to remember these stages is by the way of mnemonic. Mingling of foes mostly produces destruction. Amelogenesis can be divided into broad categories, namely pre-secretory, secretory, and post-secretory. Now let's dive into the stages. The morphogenic stage. This stage is so called because it helps determine the morphology of crown and dentino-enamel junction. The ameloblasts interact with the underlying dental papilla cells and determine the shape of the crown. In this stage, ameloblast cells appear as short and columnar. They show large oval nuclei that almost fill entire cell body. The Golgi apparatus and centrioles are located in the proximal end of the cell. Initially, mitochondria is dispersed throughout the cell, but as age advances, it occupies only the proximal aspect of the cell. Terminal bars appear at cell junction, and these terminal bars are nothing but condensation or thickening of cell membrane. Ameloblasts are separated from the underlying dental papilla by basal lamina. Below the basal lamina is a cell-free zone. The organizing stage or the differentiation stage. This stage is so called because here intracellular organelles get organized and get ready to deposit enamel. Here ameloblasts change from short columnar to tall columnar. Nucleus occupies proximal end of the cell. Golgi bodies and centrioles migrate from proximal end to the distal end, and this is called as reversal of polarity. Mitochondria can still be appreciated at the proximal end only. Cell-free zone disappears, maybe because the cells in this stage become taller. Here, ameloblasts start producing molecular signals that influence the dental papilla, which leads to the dental papilla differentiating into odontoblasts. This is a classic example of epithelial mesenchymal interaction. During the terminal phase of the organizing stage, dentine is laid down by the underlying odontoblasts. Formation of dentine here is a crucial step because initially ameloblasts derive their complete nutritional supply from the dental papilla. Now, with the deposition of dentine, this nutrient supply is cut off. Now, the ameloblasts begin receiving their nutrient supply from the blood vessels of dental sac. Moving on to the formative stage or the secretory stage. Here starts the process of reciprocal induction. For these events to take place normally, differentiating odontoblasts must receive signals from differentiating ameloblasts and vice versa, which is an example of reciprocal induction. After the first layer of dentine is deposited, ameloblasts enter formative stage. The enamel matrix formation begins. Ameloblasts remain same in shape and size, but there is appearance of blunt process at the distal end of the ameloblasts, which penetrate the basal lamina and enter into dentine, and enamel matrix formation begins. As soon as the enamel is formed, 30% mineralization occurs immediately. This stage also marks the development of Tome's process. When the ameloblasts enter into secretory phase, extensions appear into the distal end of ameloblasts, and these extensions are called as Tome's process. If we see the picture, it is clear that Tome's process is made of two parts, proximal and distal. After the enamel matrix is synthesized by ameloblasts, it is secreted through Tome's process. Once enamel matrix is synthesized, it undergoes mineralization. However, the first formed enamel does not contain any rods. As the enamel matrix synthesis continues, the Tome's process increases in their length. Remember that the rod and interrod substance is due to the presence of this Tome's process. The distal end of Tome's process secretes the enamel matrix that form enamel rods, while the proximal end of the Tome's process forms the interrod substance. Thus, rods and interrod enamel are formed. As the enamel matrix formation continues, the Tome's process disappears and the ameloblasts regain their original shape. Another layer of enamel is formed without any rods. The space previously filled by Tomes now gets filled with organic matrix, and this is now called as rod sheath. So if we recall, the first layer of enamel also lacked rods. Therefore, the final structure of enamel is composed of a rod-interrod containing layer sandwiched between thin rodless initial and final enamel layers, or in other words, structured layers sandwiched between two structureless layers. Next up, we have the maturative stage. The remaining 70% mineralization occurs here. Here, maturation or mineralization of enamel matrix occurs, and this maturation begins in cuspal or incisal edges of the developing tooth bud. The ameloblasts retain same configuration of organelles, but they come in close contact with the formed enamel. Microvilli are seen at the distal aspect of ameloblasts. Cytoplasmic vacuoles are also seen at the distal end. There is also something known as modulation that occurs during this stage. The distal end of ameloblasts shows cyclical, smooth and ruffled borders. These changes are called as modulation. This cycle occurs every 5 to 7 hours. We should also discuss about the smooth and the ruffle-ended ameloblasts. Ruffled ameloblasts have tighter distal junctions and proximal junctions are leaky, while it is opposite for the smooth-ended. The ruffled ameloblasts show numerous lysozymes in their distal ends. These lysozymes secrete proteolytic enzymes like serine proteases, and these will degrade the enamel protein and form peptides. These peptides are absorbed by the distal leaky junctions of the smooth-ended ameloblasts. Ruffled ameloblasts secrete calcium ions that help in mineralization. Also, they secrete bicarbonate, which maintains an alkaline pH at the mineralization site. Smooth-ended ameloblasts secrete water and proteins for mineralization. Moving on to the protective stage, the ameloblasts along with the stellate reticulum and oral enamel epithelium forms a protective covering over the formed enamel matrix, which is called as the reduced enamel epithelium. The reduced enamel epithelium protects newly formed enamel from coming in contact with connective tissue, failing which there will be resorption of enamel and deposition of cementum. The reduced enamel epithelium also secretes a layer of protective protein called the Nasmyth's membrane. Lastly, we have the desmolytic stage. As the name suggests, there is destruction of connective tissue above the reduced enamel epithelium for the purpose of eruption of teeth in the oral cavity. The reduced enamel epithelium releases desmolytic enzymes which break down the connective tissue, slowly leading the tooth to erupt into the oral cavity.